I repeat here the pollination. Pollination is process by which the pollen of endoecium is transferred into the stigma of gynoecium, that is the another flower or the same flower of plant bodies. So this mechanism represent the two things, the same flowers or another flowers. So it is two types. Here we represent that self-pollination and cross-pollination. What is self-pollination? And cross-pollination. What is self-pollination? When the pollen or the microspore transfer into the same flower or the another flower of the same plants is called self-pollination. It is also divided into two types, autogamy and autogamy, and another type is gate enogamy. It is here we present the pollen of same flower is fall into the stigma of the same. It is three types: homogamy, next cleistogamy, and bargamy. Homogamy, that is Catharantha species shows this type of homogamous cross pollination. One, it is also possible, the autogamy is also possible only the bisexual flowers, and here this pollen take into the stigma, that is homogamous, that is Catharantha species of plant bodies present shows this type of autogamous self-pollination, and cleistogamous pollination is present here. The beauty of species, Butia species of plant bodies, where the corolla of the flowers are not exposed in wide ranges. So it is contractile in their own bodies, and the pollen of this flower cannot overthrow or cannot spread out from this type of flowers, automatically fall into the stigma of the same flowers. So it is called cleistogamy. And bargamy, when the flower not flowerized, only positions at bud, then and the pollination is formed, then this type of autogamous pollination is called the bargamy. Gatogamy is the pollination when the flower of the same plant bodies are flowerized but not pollinated in the same flower, same flowers pollen endoeciums into the gynoeciums. The another flowers of stigma is hold the another flowers of pollen. Then this type of self-pollination is called gatogamy. Gatogamy means the neighbor, gametes means marriage. Here, self-pollination is discussed now, and the cross-pollination I repeat here. This is the pollination process where the agent is necessary. According to the function of agents, cross pollinations are three types. One, anemophily. Two, hydrophily. And three, zoophily. When the agent is air, then this type of pollination, cross pollination, is renamed as anemophily, anemophilous cross pollination, and this flower is called the anemophily flower. The rice or paddy, maize, wheat shows this type of pollinations. The features of the anemophily flowers is it is so small, it is colorless, and it is so light in weight. For these features, any type of insect or other lifeful agents cannot be necessary in this type of pollination. Only air can carry the light pollen of this type of flowers. And hydrophily, only the unisexual flowers of aquatic plant bodies can hold this type of pollinations, that is Vallisneria, that is Hydrilla, etc. The features of the hydrophilous plant bodies are the flowers are unisexuals. They can carry it by the water, so it is not the lightweight or not the heavyweight. It is not found in smell in this type of flowers, and it is not the bright in color. That is the examples of the hydrophilous plants is Hydrilla, Vallisneria, etc. Zoophily, when the agent is zoo, means animal, then this type of cross pollination is called zoophilous cross pollination, and the flower is called zoophily flowers. It is various types according to the presence of action of agents. When it is man, that is anthropophily. When it is ant, it is myrmecophily. When it is bat, it is called Chiropterophily. When it is moth, it is called Helenophily. When it is mollusk, it is Mallophily. When it is bird, it is Ornithophily. When it is butterfly, it is Zygophily, etc. And when it is insect, it is called Entomophily. So many types of these animals. Ornithophily is the Vita species, Ornithophily, man Anthropophily, ant Myrmecophily, bat Chiropterophily, etc. So there are very types of cross-pollinated plants and the agents of the cross pollinations we represent here. Next, the outbreeding devices of cross pollinations I discuss here. Outbreeding devices of cross pollination, that is diclinic, dichogamy, harpogamy, heteromorphism, and self-sterility. There are 5 types of outbreeding devices in cross-pollination. What is dicliny? Dicliny means the unisexuality. At this type of flowers, only one sexual part is present, either gynoecium or androecium. So obviously the cross pollination is situated in this type of flowers. That is pumpkin, Cucurbita maxima. Dichogamy means the maturation does not occur in the same time. That is, the maturation of reproductive part of the flowers is not occur in the same time. That is, the maturation of androecium is later, that is called protogyny, and the maturation of gynoecium is later and androecium is faster, that is called protandry. Protandry and protogyny. Harpogamy means the oppose. It is the process where the cell pollination is opposed by the structure of their sexual part of the flowers. That is, the cleistodendron, this type of organizations of sexual part, the androecium and gynoecium, is the different ways. So androecium does not fall into the stigma, it is fall into the another flower, that is heteromorphisms is the outbreeding devices of cross pollinations where the structure of the flower is the heterodactyl, that is the asymmetrical type of flowers, where any type of self pollination cannot be occur in this type of cross pollinations. Self sterility, when the androecium, the pollen of androecium is fall into the stigma of this gynoecium, it is shows the result of sterility. So this pollen cannot fulfill of these fertilizations. So it is fall into the another stigma, another flower stigma, that is called self-sterility of devices of cross pollination. So here we present the cross pollination outbreeding devices. Next, I discuss here some peculiarities of the other part of reproductive subjects, that is parthenogenesis. What is parthenogenesis? Parthenogenesis is a process by which the daughter organisms can be produced without a meeting of the male gamete and female gamete. There are two types, that is vegetative and stimulative. Vegetative and stimulative. Okay. Then naturally the parthenogenesis process is occurred in the organisms. Then this type of parthenogenesis is renamed as vegetative. That is found in the production of male bees or wasp, and already found in the plant bodies, that is Spirogyra. But when man can utilize this process by the uses of stimulant, that is colchicine, that is ultraviolet rays, etc., and produced the seedless fruits, this is called the stimulative parthenogenesis, and the utilized matter or materials, that is either chemicals or physicals, it is called the stimulant. The stimulant of parthenogenesis is colchicine, the chemical, the ultraviolet rays is called physical, etc. And I can discuss here the another part of reproduction is polyembryony. Polyembryony. What is polyembryony? When the one ovule, single ovule can produce the many numeral embryo, of the many numeral embryo is produced in a single ovule, then this type of embryony is called polyembryony. Polyembryony is two types, true and false. What is true polyembryony? True polyembryony, when the total ovule is produced many type of embryo in these ovules is called the true. But when the one ovule of a flower can produce many embryo sac, and per embryo sac can produce the many type of embryo, it's called the false polyembryony. Citrus fruits, mangoes, etc., all the flowers shows the polyembryony. Next, apomixis. What is apomixis? Apomixis is a process by which the daughter organisms can produce, besides the fertilization processes. Besides fertilization processes, the other type of organism producing processes is called apomixis. There are four types of apomixis present here. One, recurrent. Two, non-recurrent. Three, adventative. And vegetative. Adventative and vegetative. we represent the apomixis. And next I discuss the formation of endosperm. Here, endosperm, after the fertilization, the diploid endosperm is formed by the three processes, and so this process divided the endosperm as the three types, that is nuclear endosperm, cellular endosperm, and halobial endosperm. What is nuclear endosperm? The diploid endosperm here produced many type of nucleus. The central nucleus produced many type of nucleus by the cell divisions, procedures, the karyogamy. Karyogamy, or that is the division of karyokinesis, not karyogamy. And many nucleus can surrounded by the same portion of cytoplasm and produce the endosperm is called nuclear endosperm. Here the cytoplasmic division is greater than the nucleus, and many cytoplasms, a huge amount of cytoplasms can produce the cell wall, and the many cell walls covered with the cells is shows here the endosperm is the new type of different of nuclear endosperm, and halobial endosperm is the both nuclear and cellular endospermic reactions present in this type of a halobial endosperm processing. So three type of endosperms I displayed here. And next, after the reproduction, the seed is formed in the monocotyledony and dicotyledony plant bodies, and monocotyledony seeds if in here, that is the endospermic seeds, and dicotyledony seeds here the non-endospermic seeds. It is the deltoid region present in the monocotyledony seeds, and dicotyledony seeds, any type of deltoid nodule doesn't found in dicotyledony seeds. The two cotyledons present here, and the upper part is called testa and the inner part is called tegmen, and the middle part, the internal part of the seeds is called kernel. The middle division is called embryo axis, and the upper portion of the seeds where the shoot region is evolved, this is called plumule, and lower part from which the root region is ongoing is called radical. The covered layer of radical is called coleorhiza, and the cover layer of plumule is called coleoptile. It's called embryo axis, and the division in between embryo axis and plumule is called epicotyledony, and division in between embryo axis and radical is called hypocotyledony. So here we represent the two different seeds of plant bodies, that is monocotyledonous seeds and dicotyledonous seeds. Thank you so much.